Hi, my name is Corinne Weisheit, and I'm a PhD student in cellular and molecular biology at University of Michigan. Today I'm going to tell you a little bit about my thesis research. So first of all, I want you to think about all the times you have moved today. Maybe you drove to work this morning, stopped by a coffee shop on the way, you probably logged onto a computer, checked your email, maybe you even walked your dog this morning. We perform all kinds of movements throughout the day without a lot of thought. In fact, we often take for granted that in the background, our brain is working hard to take in spatial cues and direct the muscles in our body to perform these movements. For the most part, this happens rather seamlessly, and we are able to perform movements with ease. So what part of our brain is in charge of directing our movement? Well, when we take a closer look deep inside the brain, we will find a team of important structures that make up what is called the motor circuitry. This team works together to evaluate what moves need to be taken, and they send electrical signals to the appropriate muscle groups that carry out the movement of our bodies. Let's meet our team. We have the basal ganglia, the thalamus, the cerebellum, and the cortex. These structures are all connected, and when they work in harmony, they produce the basic movements that fill our day. So you can imagine that if one of the team members gets injured, the team has difficulties generating its best performance. This can result in a movement disorder. There are many types of movement disorders. Some of them you may have heard of: Parkinson's disease, Huntington's disease, essential tremor, and even Tourette's syndrome. People with movement disorders may have trouble controlling their movements. This means that parts of their body will move even when the person would very much like to remain still. People with one of these disorders may also experience difficulty executing movements. This is seen in Parkinson's patients who often have a shuffled gait. I want to take you all back to middle school health class. If you remember learning about extensors and flexors, then you're on the right track. Basically, those are the science terms that explain what happens when you extend your arm or bend your arm. What occurs is when you extend your arm, your bicep muscle should relax and your tricep should contract. And the opposite occurs when you bend your arm. So that bicep will start to contract, bringing your arm in, but this requires the relaxation of your tricep muscle. Now imagine trying to pick up something in front of you. What should happen? Well, as you reach out, the muscles in your bicep should relax and the muscle in your tricep should contract. However, if you suffer from the movement disorder DYT1 Dystonia, all of the muscles in your arm may start to contract. And these abnormal muscle contractions can spread to other muscle groups in your body, specifically your neck, your back, and even your lower extremities. Now this could distort your posture in such a way that you're no longer even facing the object that you meant to reach out and grab. Now, I told you I was going to take you back to middle school health class. Imagine you're back in class and this starts to happen to you. You're a middle schooler and you can no longer control your movements. Now, today I'm not going to try to explain how debilitating this disease can be. You can do a simple YouTube search or Google search looking for videos of dystonia and see how heart-wrenching and debilitating this disorder can be. But today I want to give you just a little bit of a glimpse at what I'm doing in the lab to study this disease in order to come up with better treatments and perhaps one day a cure. Now, I study a specific type of dystonia, but there are many kinds of dystonia. Even though most people haven't heard of this disease, it is one of the most prevalent movement disorders. It's the third most common movement disorder after essential tremor and Parkinson's disease. Although DYT1 dystonia affects muscles throughout the entire body, some types of dystonia will affect only one group of muscles. For example, cervical dystonia affects the muscles in the neck and head. This is Caitlin. She is a singer-songwriter, and she has a blog about living with dystonia. She has been a big advocate for dystonia awareness. If you have ever listened to the Diane Rehm show on NPR, you may have noticed Diane's voice is sometimes quite shaky. Diane has something called spasmodic dysphonia. This is a dystonia of the muscles that control the larynx, or the voice box. Another type of dystonia is concentrated to a very discrete subset of muscles and can affect musicians who have spent countless hours honing their skills. In this case, the repetitive practicing is what actually changes the brain, leading to these symptoms. And finally, today I'm going to talk to you about DYT1 dystonia. This is Tyler and his sister Samantha. Both were diagnosed with DYT1 dystonia when they were only 7 years old. Needless to say, their lives completely changed. However, their parents faced the adversity head-on and created the Tyler's Hope for a Dystonia Cure Research Foundation. If you want to learn more about this disease, their story, or want to know how you can support dystonia research, I would highly recommend their website. Now, what is causing these symptoms? How does this differ from something like Parkinson's disease? In Parkinson's, a part of the brain is actually degenerating, so one of our team members is losing its function. In dystonia, it is a bit more complicated than that. We are still trying to figure out exactly what is going on. But what do we know? Well, back in 1997, there was a scientific breakthrough when researchers discovered what gene was actually mutated in DYT1 dystonia. The gene, TOR1A, was the source for this mutation, and scientists determined what protein this gene encodes for and named it torsin A. Now, this protein torsin A is what is going to go out in the cell and have a job. For any genetic disorder, knowing the protein that is affected by the genetic mutation opens up a whole world of questions that scientists like me can ask in order to learn more about what is causing the disease symptoms. As a researcher studying DYT1 dystonia, I can ask, when during life is torsin A important? What job does torsin A do? Where in the body does torsin A work? And finally, how does this DYT1 mutation change torsin A's ability to do its job? Now, one scientist is not going to be able to answer all of these questions, but since this gene was identified over 15 years ago, we are getting closer to finding answers to a lot of these questions. The closer we get to answers, the closer we get to effective treatments, and perhaps one day a cure. As one example, let's address this first question. When during life is torsin A important? Because disease symptoms usually start during late childhood or early adolescence, most would assume torsin A must be important during this time in life. But that is not entirely true. The lab I work in has evidence that problems with the motor circuitry structures first begin when the structures are just being developed. We are talking about way back when the child was still in his or her mother's womb and during the first few months of infancy. This is when the genetic mutation is causing a problem, because this is when the protein is most important. As a researcher, this leads me to another question. What is going on in the brain at this time? Well, during this time in development, the neurons in the brain are migrating to their future places of work, they're making connections with their neighbors, and they also must decide which connections are important and which ones should be pruned away. This process requires a lot of orchestrated events, and it all has to take place during small windows of time. If things don't go according to plan, the motor circuitry will not be built to code. Now, if a building is not built to code, no one may notice for quite a while, until one day when a lot of stress is put on the foundation of the building and weaknesses are revealed. I believe a similar thing is going on in DYT1 dystonia brains. The motor circuitry is not built to code, but throughout childhood, everything seems fine. The child reaches expected milestones at the correct ages and is able to participate in all forms of activity without any problems. However, for reasons we do not understand, after years of use, the motor circuitry becomes stressed and begins to reveal its weaknesses. These weaknesses are observed when the wrong muscle groups are activated and the child can no longer perform this simple task that used to come easy. For my thesis research, I am trying to figure out how this genetic mutation changes the way the brain develops. Now, to address this question in the lab, I am working on developing a new tool to study this disease. I am creating a cell culture model that mimics what is going on in a developing brain. So how does this work? Well, I take neurons from the brains of mice and I migrate them to a dish. By doing this, neurons essentially have to start over in their maturation process. And after 25 days in culture, we can see that these neurons have sent out lots of projections and made connections with their neighboring neurons. They have also had to decide on which connections are important and which ones should be pruned away. This means that the same events that take place in the developing brain are taking place in our cell culture dish. By creating a cell culture model, we can study neurons at a much higher resolution, which allows for a clearer picture of what is actually making these neurons sick. I want to show you one example of this. In our lab, we have genetically engineered mice to have the DYT1 mutation that causes dystonia in humans. And when we looked at the brains of these mice, we were able to learn many things about what could be causing DYT1 dystonia. One clue is that there seems to be a buildup of waste inside the neurons. Now, by using my new cell culture tool, I can look at these problems in super high resolution. Looking at one cell at a time gives me a big advantage at studying this disease process. And what is really cool is I see the same buildup of protein waste when I looked at my neurons in culture. Importantly, from this work, we now know how to look for a sick neuron. We have to look for this waste buildup. Now, this sets me up perfectly for another important question on our list. Where in the body does torsin A work? Well, we know this protein is important in the brain, and most likely very important in the motor circuitry structures. One of those motor circuitry structures, the cortex, is the exact population of neurons that I use in my cell culture model. Now, one thing that we have known about the cortex for over 100 years is that it is made up of layers of diverse neurons. Ramon E. Cajal described this back in the 19th century when he drew the different looking cells and their layered organization. Today, we know that those different looking cells have different jobs in the brain. We also know that the layer a cell is in designates who it will talk to. So, another question I am investigating right now in the lab is whether one of the layers of the cortex is extra vulnerable to the DYT1 mutation. Or, in other words, is one layer more sick than other layers? My preliminary findings are leading me to say yes. Using my cell culture model, I am able to label different layers of the cortex and look for those sick cells with buildup of protein waste. Although this project has a long way to go, by identifying a small subset of neurons that are extra vulnerable to the DYT1 mutation, we could figure out how the motor circuitry is altered in DYT1 dystonia patients. Now, what I hope to have communicated to you is that to study DYT1 dystonia effectively, we need to look at the time during life when the mutated protein is doing its job and the place in the brain where the mutated protein is working. I am doing just this in my new cell culture model of DYT1 dystonia. Now, this project is just getting started. However, when we publish these results, other researchers around the world will be able to use this tool and ask their own questions about DYT1 dystonia. Hopefully together, we can learn more about this movement disorder and develop treatments that bring DYT1 dystonia brains up to code. Thanks for listening to my talk and watching my video. Have a great day.